Blood transfusions. Blood transfusions there's different kinds, there's different types. Whole blood transfusions are not as common as one might think. Whole blood transfusions are given when there's an overall substantial blood loss, and we need to replace that blood volume. That could be due to an injury or blood loss during surgery. Oftentimes, blood transfusions are actually partial blood transfusions given for very specific reasons, such as a packed red cell transfusion could be used to treat an individual with anemia or iron deficiency. A platelet transfusion might be given to cancer patients or other sorts of diseases that might require that. Or plasma transfusions could infuse plasma proteins needed for clotting, often used for individuals who have hemophilia. This is a short video that goes through the different blood types within the general population. If you have not watched this on your own, definitely go back and make sure that you view that. So the different human blood groups are identified by a red blood cell antigen. An antigen is a carbohydrate or a protein marker that's on the cell surface, and that identifies to the body and to other cells what kind of cell that is. We have over 30 different blood groups as humans with more than 300 different variants to it. Of all those different blood groups, the two that are the most obvious and the ones that cause the most severe reactions if they're mismatched are the ABO and the RH blood groups. These, if we mismatch, will result in severe hemolytic reactions, or also called transfusion reactions. And we don't see this severe reaction in some of these other blood groups, which is why we don't hear of them as often. In terms of the ABO blood group, there are 4 blood types based on the presence or the absence of 2 different carbohydrate antigens. These are found on the red blood cells, and depending on what antigen is or is not present on the red blood cell, that will cause unique protein antibodies to be found in the plasma that will help protect against foreign antigens. So the 2 antigens that we have in terms of the ABO blood group are the A antigen and the B antigen. Someone with type A blood has the A antigen on their red blood cells. They will then be producing anti-B antibodies in their plasma to protect against the other B antigen. On the other hand, someone with type B blood will have the B antigen on their red blood cells, and they'll be producing anti-A antibodies in their plasma. Some individuals may have both the A and the B antigen on their red blood cells. If that's the case, they will not be producing any anti-A or anti-B antibodies. And then you could be a type O individual, meaning there are no A or B antigens on the red blood cells, but these individuals will be producing both anti-A and anti-B antibodies in their plasma. In terms of blood typing, a blood sample from an individual is mixed separately with anti-A antibodies and anti-B antibodies, and then the sample is checked for agglutination. Agglutination is clumping, which means that the sample reacted with the antibodies it was exposed to. So we would put a couple of drops of blood in the A section and a couple of drops of blood in the B section. We would add the different anti-A and anti-B antibodies and then look for results. So our first sample up here, we see that when mixed with the A antibodies, we have clumping. We have no clumping with the B antibodies. So agglutination when mixed with the anti-A antibodies means that this person has type A blood. What the antibodies found were the A antigens. In the second case, the antibodies found the B antigens, so that would be an individual with B blood. In the third case, the antibodies found A and B antigens, so that would be an individual with AB blood. And in our fourth case, no antigens were found, so that would be an individual with type O blood. Hemolytic reactions, also called transfusion reactions or hemolysis, are life-threatening blood transfusion complications due to a mismatch of donor and recipient antigens. This triggers a very serious immune response. So what happens if we mismatch antigens? The donor's red blood cells will be attacked by the recipient's antibodies. So let's say you have A blood and you are given B blood, your anti-B antibodies are going to attack the B blood, and that will cause agglutination. In agglutination, the antibodies will attach to multiple cells, and those clumped cells will start to impede blood flow. Blood is not going to move through the blood vessels as readily. This diminishes the oxygen carrying capacity because now we have red blood cells that are doing their job. The agglutinated cells will then rupture. This releases hemoglobin into the bloodstream. Hemoglobin, once released from red blood cells, is toxic. And that hemoglobin accumulates in the kidneys and causes renal failure. It causes the kidneys to shut down. When the kidneys shut down, that can cause the other organs to start to shut down. And so this can result in irreparable damage to other organs and lead to death. If this happens, one of the main treatments would be to administer fluids to increase urine output. By increasing fluids, that's going to cause the kidneys to try to flush the blood and get it out of the system before it can start those toxic, irreparable effects. So the next main blood group is the Rh blood group, and this is different than the ABO blood group in a couple of different ways. The Rh blood group is abbreviation for the rhesus factor. These are two blood types based on the presence or absence of a specific protein that's on the red blood cell. So this is a different protein than what we were talking about before. So you're going to have those protein antigen markers for either AB or O blood. And then you're also going to have separate markers in regards to the Rh factor. You may contain unique protein antibodies to protect against foreign antigens in the plasma, depending on what your Rh factor is. Someone who is Rh plus has an Rh antigen on the red blood cell, but they have no anti-Rh antibodies. Someone who is Rh negative has no antigen on the red blood cell, and initially they have no anti-Rh antibodies. Their body will begin to produce anti-Rh antibodies if and when they are ever exposed to the Rh positive antigen. So that's one of the main differences from the ABO blood group. If you have, for example, type A blood, you automatically contain the anti-B antibodies. This case, if you are Rh negative, you don't have any Rh positive antibodies unless you are exposed at a certain time. The Rh factor can be typed in a similar way as we type for ABO blood group. If we mix it with the anti-Rh antibodies, there'll be no agglutination if we mix it with Rh negative. If we get agglutination, that means that we have found Rh positive blood. This is an animation just showing different blood types and blood compatibilities. So one of the unique characteristics when it comes to Rh factor is Rh sensitization and hemolytic reaction. Rh sensitization is caused when an Rh negative individual is first exposed to Rh positive blood. This does not cause a hemolytic reaction because remember, there is no preformed anti-Rh antibodies in an Rh negative person's blood. But what this sensitization causes is that Rh negative individual now starts producing anti-Rh antibodies. There's no other reactions or side effects. So this is not life-threatening, this is not harmful, but this stimulates the body to start producing those anti-Rh antibodies. So now that this person is now sensitized, any subsequent exposure to Rh positive blood will result in a life-threatening hemolytic reaction. Why this is so important has to do with pregnancy and something called erythroblastosis fetalis. This is a prenatal condition that can occur with a Rh negative mother pregnant with an Rh positive baby. Anti-Rh antibodies of an Rh negative mother could cross the placenta and attack and destroy the red blood cells of the Rh positive baby. This only occurs if the mother has been previously sensitized. So if the mother previously had a mismatched Rh positive transfusion, or if the mother had a previous pregnancy of an Rh positive baby. If this is not treated, this could result in newborn hemolytic disease in an infant after birth. So this is showing how a mother who is Rh negative in her first pregnancy could be sensitized by an Rh positive infant, and then in her second pregnancy of another Rh positive infant, then we could have the hemolytic reaction where the mother's antibodies cross the placenta and start attacking the baby. There is prevention for Rh sensitization and hemolytic reaction, and the main prevention is using the drug RhoGAM. This medication is administered through intramuscular injection to all Rh negative mothers at the 28th week of pregnancy, and then again within 72 hours after delivery. If administered correctly, this can prevent sensitization of the mother. If this does not occur or it is not caught, there are some pre-birth treatments for the infant. There could be intrauterine fetal blood transfusions before the infant's even born. And then there could be early delivery so that some of those harmful effects don't go to full manifestation. If the newborn has newborn hemolytic disease, the red blood cells of the infant will continue to be attacked after birth. The severity will vary depending on the infant. So the prognosis could be very different. It could be anemia. It could be hyperbilirubinemia, which could result in jaundice or kernicterus, like we talked about earlier, that could result in seizures and brain damage and even death. So treatments would be to give lots and lots of fluids so that the kidneys are trying to flush out some of the toxins. Phototherapy to break down the excessive bilirubin that could start to build up in the brain, and exchange blood transfusions of the infant. So here are some of the effects of erythroblastosis fidalis and newborn hemolytic disease. There are some rare blood types, and these, when we talk about blood types in terms of the 8 ABO-RH blood types, that is an oversimplification. When we talk about the 8 ABO or RH blood types, that's an oversimplification. When we talk about the 8 ABO-RH blood groups, that is an oversimplification. We actually have up to 342 different antigens on our red blood cells, which means there are millions of different blood types out there. Because there's over 30 different blood systems, each can be subdivided, and so we can have over 300 variants of these different types of blood. The reason we talk about ABO-RH so much is because those cause the most severe hemolytic reactions, but there are many other types. Rare blood types would be blood types where an individual lacks an antigen that most people have, or they have an antigen that most people don't have. A good example of this would be in terms of the RH system. So we talk about RH positive and RH negative, but the RH system actually has 61 different antigens. When we talk about RH positive, RH negative, that's only one of those 61 antigens, and that would be antigen D. Nearly everyone has at least one, and usually more, of those 61 RH antigens. And so we just simplify it by saying RH positive, RH negative. Okay. But there is such a thing as the golden blood, and this would be RH null. Now, this isn't actually blood that's golden in color, but it's called golden blood because it's so rare. Golden blood, or someone who is RH null, lacks all 61 antigens. This is the rarest blood on Earth. Since it was discovered in 1961, there's only 43 people who have been documented having this blood. Currently, there are only 9 active donors worldwide that has this blood. And so this is extremely rare.